Greetings. Welcome to the MSA Coalition 2021 Annual Patient and Family Conference, brought to you virtually again this year as the impact of the global pandemic continues. Thank you for joining us. I'm Cyndi Roemer, Chair of the MSA Coalition. I wish that I could be at the registration desk, greeting you each individually, sharing a hug with old friends, that I could be standing at the podium welcoming you as I look out and see both new and familiar faces. Hopefully that will be possible next year. In the interim, I'm grateful to all those who have made it possible to deliver this conference virtually. For over 30 years, the MSA Coalition has been providing support and education through this program. So I'm pleased that we've been able to continue to provide this valuable resource virtually these past 2 years. As a former care partner myself, I know how critical it is to have much needed support and accurate medical information to effectively navigate life with MSA. Though virtual, our amazing conference committee and event staff have put together an outstanding program that includes opportunities to connect with others and learn from the experts, including the chance to ask questions. You have already had the opportunity to participate in our pre-conference activities, learning about the programs and services the MSA Coalition provides, attending support group meetings, and gaining considerable education and inspiration from members of our team, as well as our charity partners from around the world. Next, you will continue to learn about the medical aspects of MSA, get updates on current research, and take advantage of support activities as you interact with others also living with MSA. We are confident there is something in this program for everyone. So please join me in thanking all those who have made this conference possible. Of course, our sponsors. Our gold sponsor, Biohaven Pharmaceuticals. Our silver sponsor, Lundbeck. And silver sponsor, Theravance Biopharma. And our bronze sponsor, Alterity Therapeutics. We appreciate our charity partners who are offering sessions delivered by members of their organizations. MSA Trust, the Bishop Karl Gulser Foundation, Aramis, MSA Belgium, MSA New Jersey, and the Brain Support Network. Please do visit their booths to personally connect and thank them for their participation and support. To all our presenters, volunteers, and staff, without whom this conference would not be possible, many, many thanks. And of course, a big thanks to our medical host team at the Mayo Clinic. All members of the team, especially renowned MSA researcher, Dr. Philip Lowe, Dr. Elizabeth Kuhn, recipient of the MSA Coalition Don Summers Travel Award, and Dr. Wolfgang Singer, a longtime friend who always provides support when asked and who has coordinated this great team at the Mayo Clinic. It is my pleasure now to introduce you to Dr. Philip Lowe and Dr. Wolfgang Singer. Dr. Singer is an established neurologist receiving accolades among patients for his thorough and caring approach to treatment. He is a well-recognized investigator among the MSA research community. Dr. Singer began his career in education in Nuremberg, then completed his internship and residency at the Mayo Clinic before joining the faculty. There, he was mentored by Dr. Philip Lowe, also a neurologist at the Mayo Clinic. Dr. Lowe is a pioneer in MSA research. His work over the years is well known. He has published significantly on MSA and received several awards over the years for his work in neurology. We had the pleasure of spending some time with Dr. Lowe and Dr. Singer as they look back and look ahead in a conversation between these two outstanding professionals with a shared history. Thank you.
So here we are for the fireplace chat in a virtual world. Not really a fireplace chat, but we can at least make it look that way. There we we are. And we were invited to kind of discuss a little bit the Mayo program, our interest in multiple system atrophy, where it came from, where it will hopefully go in the future, and reflect a little bit on the experience in MSA trials over the last years at the site. So I'm here with Philip Lowe, who has been my mentor for, I think it's almost 2 decades now, right? And Philip Lowe came here to Mayo, originally from Fiji, and he can tell us a little bit more about how he got here and how he developed the autonomic program that ultimately made us interested in multiple system atrophy.
Thank you, Wolfgang. I developed my interest in Australia, where after graduation, I learned something on testing of baroreflexes, got me interested in the autonomic nervous system. And I came over to Mayo, and one thing led to another, and this led to the development of the autonomic laboratory, because it had always been regarded that the autonomic nervous system was the Cinderella of medicine. It was Cinderella because it was neglected. There were methods to test it, but not clinically. So this led to the development of the autonomic lab in 1983. And on the research side, we persevered, and after the necessary tools, we developed the first Autonomic Disorders Program Project. And this merged into the Msa Program Project. The Msa Program Project was started by Cliff Schultz, who was interested in an MSA treatment trial. Unfortunately, he did not survive. So he asked me to take over the program, and the program focused on the natural history of MSA. And this led eventually to a treatment trial. We based our scientific basis on some experimental data done on the animal model of MSA, and that seemed to suggest that rifampicin might be effective. So I headed an MSA randomized treatment trial. Now, unfortunately, that was negative, but it did provide us with a good starting point for our program on multiple system atrophy.
Yeah, and I think it was just before the rifampicin treatment trial started out that I came from Germany as a green first-year resident and was taken under your wings, and you taught me everything there is to know about the autonomic nervous system, and triggered my interest in autonomic disorders and multiple system atrophy. Back then, Philip Lowe, I remember, he ran this trial basically by himself. He saw every single patient, he scored every single patient, which really was a role model in terms of involvement of an investigator in a treatment trial like this. So what became what was originally a year of research that was supposed to spend with Philip Lowe became years, and eventually I decided to pursue my career here in the United States, and did the training here at Mayo, and then joined Philip Lowe's program, where we have been collaborating now for many years on various projects. I started out helping with the rifampicin treatment trial, and from there we went to our stem cell research that really sort of popped up after the negative rifampicin treatment trial, where human-grade mesenchymal stem cells were produced at the Mayo Clinic, and we had the opportunity to connect with the head of the stem cell lab here, who we had discussions with, and it became clear that there was clearly an application for multiple system atrophy with these cells, which produce growth factors that we know are missing in multiple system atrophy. And we went ahead with the early phase 1 or phase 1/2 treatment trial of mesenchymal stem cells in multiple system atrophy.
Yes, Wolfgang, I think that it might be worthwhile to spend a moment thinking about the basis for stem cell. And this was based on observations that were made that there's a growth factor deficiency, and in particular BDNF and GDNF and NGF. But it's likely, don't you think, that what it really does, apart from correcting the deficiency, is that it alters the microenvironment of the neuron so that it sustains a more normal environment.
Right, right. I do think that there's lots of unknowns really about how these stem cells can positively affect a disease course in a neurodegenerative disease, and it's much more complicated than we believe it to be. The growth factor deficiency and the ability of these cells to produce exactly those growth factors is intriguing, but I think quite likely not the only mechanism.
So what's the most positive findings that you have made with the studies so far on stem cell?
Right. Well, I think at first it was very surprising actually to me to see how well patients did that were exposed to that treatment. Much more than I had experienced before and what I knew about the natural history of multiple system atrophy. So that was the first sort of observation just doing the trial. But then there were some additional findings that really corroborated sort of this initial impression. One, that there seemed to be a dose-dependent effect. There was an ideal dose for patients really did very well with, and much higher or much lower doses seemed to be less effective. And then as the work progressed, finding biomarker support that we are indeed increasing the neurotrophic factors in the spinal fluid that we could measure, and then also that the marker of neuronal or nerve cell degeneration, neurofilament light chain, actually decreased in patients who were treated with mesenchymal stem cells. Were all supporting the initial impression.
Yeah, for those of you that don't know too much about neurofilament light chain or NFL, this is actually a measurement of the axon of neurons. But what happens as the neuron breaks down, the axon also breaks down. So it's a very good index of central neuronal degeneration. So what Wolfgang has referred to is one of the few studies, in fact, the only study I can think of where there is demonstration that you have reduced the rate of neuronal degeneration. So that's actually very exciting. And on the clinical side, perhaps Wolfgang can tell us a little about the patients that have continued to get treatment after the 1 year.
Right. So after those initial positive signals, we were faced with the dilemma, which really wasn't a dilemma, but it initially presented itself that way, that patients who completed the 1 year trial wanted additional treatments. And we approached the FDA and received permission to continue to treat patients who were in the original phase 1/2 trial on an ongoing basis if they chose to be part of it. That took a little while to get that approval and get the logistics ready. So a number of patients had already been without treatment for a year or a year and a half, and some of them in that time frame progressed considerably and were no longer really eligible to be included. But all patients that directly were able to go from the original trial to the extension to the continued extension chose to do that. So 16 of the original 24 patients did that, and some of them have received every 6 month injections now for almost 5 years.
See, that is very interesting because I know of no treatment in MSA that have sustained the patient for as long as 5 years. That's truly exciting.
Right.
And that really has excited not only investigators at Mayo, but outside of Mayo, and also at the FDA. And perhaps Wolfgang could tell us a little about some exciting news he has obtained about a new study in mesenchymal stem cell.
Yeah, so it was a long process and a lot of work to create and design the next phase trial that was exciting enough for the federal support system to be willing to support such an expensive and ambitious trial. And we just literally 3 days ago found out that the next phase trial of mesenchymal stem cells in multiple system atrophy will be funded and is slated to start later this year. This will be a trial where we explore. We're pretty comfortable that we have the right dose at this point. We just don't know really the optimal treatment frequency. Should it be every 3 months? Should it be every 6 months? So we'll explore that in an initial phase and then move on with the best treatment that will be defined in an interim analysis. And we'll compare that to placebo, which we'll have to do in order to provide the necessary proof for regulatory agencies that this is really a treatment that works.
I thought from the patient standpoint, although it's placebo-controlled, I thought it was interesting that there is a 2-to-1 balance of patient versus placebo.
Right. And that was done on purpose. We wanted to increase the chance of patients to get the active treatment and still be able to compare to patients who do not get that treatment. And so by assigning 2 out of 3 patients to the active treatment was the compromise, and I think is a good compromise and makes it just more desirable for a patient to be part of this trial and have a better chance to be actually in an active treatment arm.
And the good news is that the study is going to start reasonably soon. Is that right?
That's right. That's right. We're hoping later this year. We obviously have now some final preparatory work to do before the study can actually get started. But over the next couple of months, I think we should be ready to go ahead.
You know, it's important to know that stem cell is not the only approach. I want to spend a moment pointing out that it's sad but true that except for stem cell, every study so far has been negative. And one of the reasons, I've been thinking about that quite a bit, and one of the reasons, of course, is that if you look at certain processes, whether it's in inflammation, oxidative stress, mitochondria, etc., it occurs in pretty much all the degenerative diseases. And until you find the switch, the rate limiting step, if you focus on that and you stop that, then things go very well. A good example is a condition called TTR amyloid. Treatment trials on that have been dramatically positive because they have found the rate limiting step. Perhaps Wolfgang can tell us a little about what we think might be the rate limiting step in MSA.
Yeah, you really put me on the spot there because I don't think we know. I think there's a lot of focus on alpha-synuclein and alpha-synuclein misfolding, as this is the pathologic substrate. That's what we find in the brain of patients with multiple system atrophy in excess and in abnormally configured form. But do we really know that this is ultimately the cause for the disease or for the progression of the disease? I don't think we can say that.
I think Wolfgang has expressed it very well. It's always good to have some scientific skepticism. Currently, the scientific community has put all their money onto this presumed step. And let's spend a moment on this step again. This is a step where the alpha-synuclein monomer becomes the toxic oligomer. And the things that can go wrong are, one, there is evidence that there's too much production. There's secondly, evidence that there is a garbage disposal sort of mechanism called autophagocytosis or autophagy that could be defective. And the third step is that the spread from neuron to neuron is excessive. So perhaps we can explore a little bit on each of those steps. The first is production. Any thoughts on production and what's been done about it?
Yeah. Well, the idea is really to limit the production of alpha-synuclein in general, right? So there's really not really a focus on the misfolded alpha-synuclein. It's just the production of alpha-synuclein in general with genetic approaches.
That's true, although there is some attempts to make it a little bit more differential. For instance, with the vaccine, the configuration of the vaccine is hoped to do a little bit more with blocking the oligomer.
Right.
So a second step is the idea of increasing autophagy.
Right.
Any thoughts on that?
So the idea of autophagocytosis and enhancing that part is an interesting one. I think we don't really know how much of a key role the effect in that area is actually playing in the disease. There is an ongoing, or now I think completed trial at NYU looking at sirolimus to enhance autophagocytosis. And I believe that study is currently being looked at. We'll probably find soon out about the outcome. And of course, then as a next step, there's the mechanism of approaching the alpha-synuclein, misfolded alpha-synuclein that's already there with antibodies. So what are your thoughts on that?
Well, as you know, monoclonal antibodies can be directed against a target, and it can be very effective. It's been used in the treatment of COVID. As you know, the past president, when he had COVID, received monoclonal antibody intravenously, and it worked very well. So it's a logical step to focus that on the oligomer. And there quite a few different companies have produced different monoclonal antibodies directed against the oligomer.
Right.
I think the major problem is accessing the brain. There is the blood-brain barrier, and something you give intravenously will not get across. This is not completely true in that it is considered that in MSA, the blood-brain barrier is partially defective. So the hope is that you could get a certain amount into it. And there is a study that's going to be started, hopefully within this year, by Lundbeck, I think, which is a monoclonal antibody. And the hope is that you'd get sufficient across to access it.
Right.
A different approach, of course, would be to give it intrathecally or even more directly into the ventricular space.
Right, right. And that certainly would increase the availability within the central nervous system. But there would still be a little bit concerned about the fact that these antibodies are not making it into the cell, right? So really, in theory, what they would do is they would block the cell-to-cell spread, the prion-like spread that has been described, but not attack already existing intracellular alpha-synuclein deposits.
I think, Wolfgang, you are discussing a very difficult problem. When you're dealing with a molecule that is large enough, like the monoclonal antibody, it's almost impossible to get it across perfectly well. You can try various vectors. You can try things like the adenoviral vector, but that has trouble with large proteins also.
Right.
So it's a very difficult problem.
Yeah.
And that's the main reason why this approach has not been that successful so far.
Yeah. Well, let's hope that will change, and that interfering with the cell-to-cell spread is enough to make a difference in disease progression. But that topic of large molecules and their difficulties in a disease like multiple system atrophy kind of gets us to the idea of these small molecules that have been developed to prevent or reduce the misfolding of alpha-synuclein. But what are your thoughts on some of those developments?
I think that it is very exciting. Some of these, one in particular, can be taken orally, and the molecule is small enough to get to where you need to go. And the big question, of course, is this truly the rate-limiting step?
Right.
We hope it is, because there's been years of work put into it.
Right.
You know, the approaches we have, you need to identify the rate-limiting step. You need to get a drug that gets to where it needs to go. And then you get to try to get it there at an early enough stage, which leads us to a question of what about getting to the disease at an early stage? Wolfgang has been working very hard on the question of looking at prodromal or even preclinical forms of MSA. And tell us a little about your work there.
Yeah, of course, this is a topic that would be sort of the ultimate solution, the ultimate way of addressing this, by treating a disease before it's actually manifests, before it's actually causing symptoms. And what we have been working on over the last few years now is to look at markers that would identify multiple system atrophy and related conditions at an early stage. And we have found that a technique called PMCA, or protein misfolding cyclic amplification, allows us to detect the misfolded oligomer that is specific for multiple system atrophy very early on in the disease course, in early multiple system atrophy, and that a different type actually of misfolded alpha-synuclein is detectable in early cases of Parkinson's disease and dementia with Lewy bodies, in that this marker very nicely, basically perfectly separates those groups. And in addition to that, another marker, you mentioned before, neurofilament light chain, is another marker that is very high in the very early stages of multiple system atrophy, and much lower in related conditions. And those two markers combined really seem to provide an almost perfect separation between early stages of multiple system atrophy and potential mimickers, which certainly would allow us to make a diagnosis much more certain at a much earlier stage.
Yes, you know, I think Wolfgang has pointed out this difference between MSA and the Lewy body diseases like Parkinson's disease. As the audience knows, MSA is really not Parkinson's disease on steroids, because the biomarker or the oligomer that is present in MSA is present in glia, whereas the one in Parkinson's disease is present within the neuron. And in a recent study that we part of also done by Dr. Soto and group, they showed that the structure of the oligomer in MSA and that of Parkinson's disease is structurally different. And that has led to this biomarker. Now that you've got the biomarker that separates the two, tell us a little about how you can look at the early forms.
Yeah. So that was the original studies in multiple system atrophy and Parkinson's disease. And then we thought that we could look at an earlier stage, because as you know, many patients with multiple system atrophy present with predominantly autonomic problems, orthostatic hypotension or bladder symptoms. And at that stage, it is clinically really impossible to differentiate a patient who has autonomic failure alone or who has autonomic failure as an early sign of multiple system atrophy. And so we looked at a group of patients with isolated autonomic failure, we call them pure autonomic failure, and collected these markers at a stage where they only had autonomic problems, and followed patients over the next few years, and found that we were able to predict with this marker which patients will go on to develop multiple system atrophy and who do not. So there we are at a stage where patients do not have any problems with movement, with coordination, with anything other than the autonomic nervous system, and we are able to identify patients at that stage. Now, wouldn't it be nice to go even earlier than that, right? Where patients have only sleep problems. Many of you probably know about the term REM sleep behavior disorder, or may even be suffering from it. And it is not uncommon, or actually more common than not, to develop that before anything else, before even autonomic symptoms are present. And at a stage where you only act out your dreams, we potentially could detect if you are destined to develop multiple system atrophy. And this is something we're currently testing in a NIH-funded study that will be going on for the next few years.
You know, this approach of recognizing MSA before you even develop full-form MSA is very important, especially when you have effective treatment. But for those patients who have either PAF or REM sleep behavior, it's important to point out that most of you will not develop MSA.
Right.
Perhaps no more than 25% of PAF will develop MSA. And as far as REM sleep behavior is concerned, we don't know the percentage yet, but it's very low. It's probably no more than 5%, but we don't know.
Right, right. And this is part of what we'll try to find out.
Yeah.
Right. Now, switching topic, I wonder if we should look at what the next few years may bring, may hold. And there's something that's sort of immediately on the horizon that I'm sure all MSA patients are very much interested in at the moment, which is the myeloperoxidase inhibitor studied by Biohaven. I think with this study, we are very close to finding out the results of a large phase 3 trial. I understand the results are expected later this year yet. And I would be curious what your thoughts are, what your expectations are on this.
I think that blocking inflammation is important, especially inflammation associated with microglia. It's this $64 question of whether it's a rate limiting step or not.
Right.
I think the good news here is that we are so close to the results. Let's wait and see what it shows. There is some rationale about this study, and that rationale is based on a small study done on 40-odd patients who were given the drug for 3 months, there's a control group and there is a treatment group, and there was some separation. The numbers are too small to look at significance, but at least it provides some basis for it.
Right, right. So let's hope for the best. It would
Yeah.
be nice to have a treatment, at least some positive effect seen in that study, and we will see.
And it is on a accelerated approval pathway, meaning that doesn't mean that the drug has to work. What it means is that the FDA recognizes that MSA is a nasty disease. They're desperately trying to help us find something, and that if the drug does work, then they will try to get it approved more rapidly.
Right.
That's one approach. The other approaches, as we mentioned, there is the monoclonal antibody study.
Right.
There is the stem cell study. We should provide a word of caution, is that based on what Wolfgang has found early on, stem cell will likely only help for patients that are fairly early on in the disease, so that as the disease progresses, it probably won't help. So we don't want to be over optimistic about that.
Right. But I do think a number of approaches are either imminent to be studied, or at least on the horizon to be studied over the next year or two. And I would say it's really exciting times ahead, where we're not struggling to find one new treatment. We are having actually competing enrollment into treatment trials, which is a wonderful situation to be in. And I do think that we'll hopefully see some early efficacy signals in some of those.
Yes, there's a good number of treatment trials that's on the horizon. The scientific community is very much in keeping with patients in recognizing the urgency of the problem. And that has led us to finding quicker methods of looking at drugs that are relevant. And one of the approaches that we have done is to look at drugs that are potentially effective for MSA that has been approved for different diseases. So say you look at a certain mechanism, then you find drugs that have been explored and approved for other conditions. That way we can get approval to study it without going through a lot of preliminary studies, so we can get that working in a trial in patients fairly early.
Right. And that was the strategy with rifampicin, for example, right? That was already approved for treating infectious diseases. And we are also seeing that with the new drugs that are addressing the glucose metabolism.
Right.
That are also an exciting area that will be further explored.
Right. There's good evidence that there's insulin resistance in not only patients with diabetes, but the same process occurs in the brain of MSA patients.
Right, right. Well, I would say with that, we close our fireside talk, and we look forward to interacting with you throughout the rest of this meeting, and we'll be there for other sessions to answer questions. And we thank you for your attention.
Thank you.